Everyone, this is our first topic, biomolecules classification, types, carbohydrates, lipids, proteins and fats. Biomolecules refers to any molecule that is produced by living organism. जो भी molecule living organism तैयार कर रहा है, that is called biomolecule. As such, most of them are organic molecules. The four major groups of biomolecules include polysaccharides, proteins, nucleic acids, and lipids. They are found in and produced by living organisms. Thus, many of the biomolecules are polymers. A polymer is a compound made up of several repeating units or protomers and produced by polymerization. So most of these biomolecules are organic compounds. Being organic means in general they contain carbon atoms. इनके बीच में दो एटम्स के बीच में कोवेलेंट बॉन्ड होगा, कोवेलेंटली बाउंड टू अदर एटम्स, especially carbon-carbon and carbon-hydrogen bond. So the four major element constituents are carbon, hydrogen, oxygen and nitrogen. Chemicals or biomolecules present in the living organism, ये हमने स्टार्टिंग में देखा, these are called biomolecules. They come in a wide range of composition and sizes. Carbohydrates, lipids, nucleic acids and proteins are four main types of biomolecules. Biomolecules are divided into two categories, inorganic and organic. Inorganic biomolecules include minerals, gases and water, and organic biomolecules include carbohydrates, protein, fats, nucleic acids, vitamins, etc. Different biomolecules can be classified as aldehyde group, ketones, and aromatic compounds as chemical forms. The amino acid, nucleotides, and fatty acids can be classified as biochemical forms. Carbohydrates are made up of carbon, hydrogen and oxygen. ये सब लोग जानते हैं. Hence, carbohydrates like cellulose, starch, glycogen, glucose, fructose, etc. are the most abundant biomolecules on Earth. ये maximum quantity में अर्थ पे मिल रहे हैं देखने के लिए. There are three types of carbohydrate, तीन तरह के होंगे, mono, di and poly. First one is monosaccharide. These are the simplest of carbohydrate, simplest form, and are known as sugars. They are the building units of complex carbohydrates. These cannot be hydrolyzed. Figure of monosaccharides: glucose, fructose, galactose. Oligosaccharide: these consist of more than one but fewer number of monosaccharides. एक से ज्यादा होगा पर बहुत ज्यादा नहीं होंगे. Like polysaccharides में तो बहुत सारे होते हैं, इसमें only fewer. Joined together by glycosidic bonds. Glycosidic bond हमें याद रखना है, क्वेश्चन आता है नेट में, स्लेट में भी मैंने लिखा हुआ देखा है. ये दोनों के बीच में कौन सा बॉन्ड है? दिस इज glycosidic bond. On hydrolysis, they yield the monosaccharides units which may be similar or dissimilar. Third one is polysaccharide. These consist of a large number of, often thousands. अब दोनों में डिफरेंस क्लियर हो गया होगा. ऑलिगोसैकेराइड्स में एक या दो या चार जो भी कुछ है, और पॉलीसैकेराइड में जो नंबर होगा थाउजेंड्स की फॉर्म में. These consist of a large number of monosaccharide units to form branched or unbranched chains. These can be hydrolyzed to yield monosaccharides units which are usually similar. The structure of monosaccharides: glucose and starch. Next one is proteins. Proteins are organic nitrogenous compounds. in which a large number of amino acids are joined together by peptide linkage or peptide bonds to form long polypeptide chain. एक लंबी चेन बन जाती है प्रोटीन बनाने के लिए, जिसमें बहुत सारे अमीनो एसिड्स जुड़े हुए। स्ट्रक्चर ऑफ प्रोटीन्स सभी लोग पढ़ चुके हैं इसको। 4 टाइप्स होंगे इसके। प्राइमरी स्ट्रक्चर, it is the basic structure of a protein in which a number of polypeptides are involved, having a sequence of amino acids. The first amino acid of the sequence is called N-terminal amino acid, and the last amino acid of the peptide chain is called the C-terminal amino acid. Second form is secondary structure. Secondary structure protein threads form a helix. There are 3 types of secondary structure: alpha helix, beta pleated, and collagen. In alpha helix, the polypeptide chain is coiled spirally in a right-handed manner. In beta pleated secondary proteins, 2 or more polypeptide chains are interconnected by hydrogen bonds. In collagen, there are 3 strands or polypeptide coiled around one another by hydrogen bond. तीनों में आप डिफरेंशिएट कर सकते हैं, अल्फा, बीटा एंड कोलेजन. Third one is tertiary structure. The long protein chain is folded upon itself like a hollow woolen ball to give a 3-dimensional view of proteins. Last one is quaternary structure. Each polypeptide develops its own tertiary structure and functions as a subunit of protein. Example, hemoglobin. So please correct the spelling of hemoglobin, HAE. A is missing. तो इसको ठीक कर लीजिएगा. In the adult, 4 human subunits of hemoglobin are involved. The 2 subunits are of alpha type and 2 subunits of beta types. The structure: primary structure, secondary structure, tertiary and quaternary structure of protein molecules. म्यूटारोटेशन एक न्यू वर्ड है जो इससे रिलेटेड क्वेश्चंस लगातार आते रहे हैं नेट पे. So म्यूटारोटेशन was discovered by Davenport in 1844. He noticed that there was a change in the specific rotation of sugar. एक इनको एक स्पेशल टाइप का एक चेंज मिला, कुछ स्पेसिफिक रोटेशन मिले in an aqueous solution with respect to time, that is called mutarotation. It is a deviation from a specific rotation due to the change in the equilibrium between alpha anomeric and beta anomeric form in the aqueous solution. For example, mutarotation is present in the glucose molecule structure, and this is the mutarotation equilibrium. To remember, जो हमें याद रखना है, glycosidic bonds are covalent chemical bonds that hold together by a glycoside. Glycoside क्या है? A glycoside is simply a ring-shaped sugar molecule that is attached to another molecule. A glycosidic bond forms by condensation reaction, which means that one water molecule is produced during the formation of a glycoside. This is called condensation. Next one is amino acids. ये सभी लोग जानते हैं 20 amino acids हैं, 10th क्लास से हम लोग पढ़ते आ रहे हैं. Amino acids are the building blocks of proteins. There are thousands of amino acids available in nature. Protein contains only 20 different kinds of amino acids, all of which are alpha amino acids. The same 20 standard amino acids make proteins in all the living cells. May it either be a virus, yeast, bacteria, plant or human cell. These 20 amino acids combine in different sequences and numbers to form various proteins. Types of amino acids based on charge. कुछ में नेगेटिव चार्ज होगा, कुछ में पॉजिटिव होगा, कहीं पे चार्ज ही नहीं होगा. So on the basis of charges, amino acids can be classified as follows. Acidic amino acids. Acidic amino acids that contain a negative charge or an acidic group are called acidic amino acid. Example, aspartic acid, glutamic acid. Structure of aspartic acid and glutamic acid. You can see negative. Basic amino acids that contain a positive charge or a basic group basic amino acids. Example, lysine, arginine or histidine. Of lysine, arginine and histidine. Neutral amino acids, नाम से समझ आ रहा है कोई चार्ज नहीं होगा. Those amino acids that do not contain any charge on the R group are called neutral amino acids. Example, valine, glycine, alanine. The structure of glycine, valine and alanine. Types of amino acids on the basis of nutrition. On the basis of nutrition, amino acids can be classified as follows. Essential amino acids. These amino acids cannot be synthesized in the animal body and therefore must be present in their diet. Required. जो ऐसे amino acids हमारे body में नहीं बन रहे, those are called essential. Example, in human beings, essential amino acids are valine, isoleucine, phenylalanine, methionine, leucine, lysine, tryptophan and threonine. नॉन एसेंशियल अमीनो एसिड्स जो बॉडी में ऑलरेडी बन रहे हैं, जरूरत नहीं है हमको बाहर से लेने की. These amino acids can be synthesized in the animal body and may not be supplied in the diet. For example, alanine, arginine, tyrosine, asparagine, glutamine, proline, etc. In plants, all the amino acids are non-essential. याद रखिएगा इसको. In plants, all the amino acids are non-essential. For example, in human beings, essential amino acids are valine, isoleucine, phenylalanine, methionine, leucine, lysine, tryptophan and threonine. Remember as VIP, MLLTT as a sequence, for example, of essential amino acids. Effects of pH and temperature on protein. Each protein shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. जो इसका optimum temperature है, कम या ज्यादा होगा, इसकी एक्टिविटी कम हो जाएगी. Low temperature preserves the enzyme in a temporary inactive state. अगर कम टेम्परेचर पर रखेंगे तो ये इनएक्टिव हो जाएंगे. Whereas high temperature destroys enzymatic activity because you know that proteins are denatured by heat. Fats are a subgroup of compounds known as lipids that are found in the body and have the general property of being hydrophobic. They are insoluble in water. आपने वर्ड सुना है, phobic like phobia, किसी चीज से डरना, water hating, and philic means loving. So these are hydrophobic. Fats are also termed triglycerides molecules made from the combination of one molecule of glycerol with three fatty acids. Fatty acids क्या है? A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl or ethyl or higher number of CH2 group, it means 1 carbon to 19 carbon atoms. Then glycerol, it has 3 carbon atoms, each of which has a hydroxyl group bound to it. It is a simple polyol compound. It is colorless, odorless, viscous liquid that is sweet tasting and non-toxic. Difference between saturated and unsaturated fatty acids, meaning Saturated fatty acids contain a single chain of carbon atoms with no double bond. One unsaturated, these contains carbon chain with one or more double bonds. Second difference is hydrocarbon chain without double bond, only single bond. Hydrocarbon chain with one or more double bonds. Third is physical appearance, solid at room temperature but unsaturated, liquid at room temperature. Type of chain, in saturated fatty acids, straight chain is found, unsaturated, bent chain at double bond. Melting point of saturated fatty acids relatively higher, but in unsaturated relatively lower. Sources to obtain saturated fatty acids you can obtain from animal fats, palm oil, etc. and unsaturated from plant and vegetable oil, avocado, sunflower oil, walnut, etc. Shelf life, they do not get spoiled quickly and in unsaturated they get spoiled quickly. Types of fatty acids, three types: monoglyceride, di and triglycerides. In monoglyceride, condensation of one fatty acid and glycerol. In di, condensation of two fatty acids and glycerol. Third is triglyceride, condensation of three fatty acids and glycerol. Structure of mono, di and triglycerides. To remember, ester bonds in a fat molecule, the fatty acids are attached to each of the three carbon of the glycerol molecule with an ester bond through the oxygen atom. Usually, esters are derived from carboxylic acid and alcohol. Here are the questions you have to write down in your notebook. What are carbohydrates made up of? What are the example of inorganic biomolecules? What are the example of organic biomolecules? What are the example of essential amino acids? And what are the example of non-essential amino acids? Just revise it. We will meet again tomorrow. All the best.